Welcome geniuses, I'm Genie, your best buddy for A-levels. In this channel, we'll bring you to explore the secret formula behind success.
Okay, today I'm going to cover AS topic, topic 7, transport in plants. I'm going to focus on learning outcome 7.2G. You're going to see how assimilates such as sucrose and amino acids are transported from the source into the sieve tube, commonly known as active loading of sucrose from the source into the sieve tube. So now I'm going to see how a phloem tissue looks like before we look at what is actually the process of loading. So we need to identify who is the sieve tube and who is the source. So I'm going to draw. Okay, but before that, let's look at the example of questions so that we know what to prepare and what to focus on. So you see that in MCQ, they will like to ask you very detailed questions like what is the transport that is responsible in transporting the sucrose from the source, which is like your leaf that is responsible of making the sucrose, and how does it enter the companion cell before they are being sent into the sieve tube. So another similar question here that we have. And for structured question, sometimes they will be guided in the sense that diagrams are given to you and is more detailed because they would want you to describe based on the question itself. So they will give you something like that. But if you wouldn't know the the rough idea on the arrangement of the cells, so even if you're given this, you might not be able to relate where is the inside and where is the outside, where is the sucrose being made and where they're transported to. So here, I will draw a simplified diagram and guide you on how to identify the position and the movement. So now I'm going to start with the process of active loading, but before that, just an intro about the structure. So we know that actually phloem is actually a tissue. And this particular tissue is made up of sieve tube, sometimes some call it sieve tube element, and companion cell. So these two types of cell will form your phloem tissue. So under light microscope, how would you identify? Usually xylem is a larger cell with a hollow lumen, but for a companion cell, they are relatively smaller. They have a very thin wall. And the most prominent feature here is they actually comes with a smaller cell adjacent to it, known as the companion cell. And the companion cell usually is a cell that's very dense, not only has a nucleus, it has lots of mitochondria to produce a lot of ATP for the process of loading of sucrose. And as for the sieve tube, then how do you distinguish it from the xylem vessel? Phloem sieve tube element, they don't have a nucleus, but they do have little cytoplasm. Another feature of the sieve tube element is it still has a end wall, but in order to reduce resistance for the transport of sucrose, they actually make pores on the wall known as sieve pores. And this structure is actually known as sieve plate. So this is actually the cross-sectional view of the phloem. And what we're going to show later here is the longitudinal section of the phloem, where you will see how the sucrose is being loaded from the source into the sieve tube. So here I'm going to draw the longitudinal section. So this will be the source cell. And then I'll show a neighboring companion cell. And next to the companion cell, you have your sieve tube. So you have the sieve tube. This will be the source cell. Companion cell. And next to the sieve tube, a vessel, a xylem vessel, because in a vascular bundle, usually next to your phloem, you have your xylem. So this is the longitudinal section. So where is the sucrose being made now? We're going to see how the sucrose that is made in the source cell that is going to be transported into the companion cell before they are being loaded into the sieve tube. So from here, we can see how it enters the companion cell and how it needs to leave the sieve tube. So from the look of it, if you want to get, if they can come up with a 4 marks question, so obviously it's not as easy as you think. So we need to look into the detail. So now we're going to draw the details within the wall. Okay, I'm not drawing to scale because I need to focus on what is happening within the wall. Okay, so this will be the cell wall. So how does the sucrose enter the companion cell? It's a bit complicated because they don't go in by their own, where it's like normal facilitated diffusion using a carrier protein. They need another ion to bring them in. So when two molecules need to enter or go through a transport protein together, they are known as co-transporter. So who does the sucrose need to be co-transported with? It's with another ion known as H+. So where does the plant cells get the H+ from? So the H+ is actually accumulated in the cell wall where they are actually being pumped from the companion cell. So the accumulation of the H+ actually is coming from the companion cell. And how do they create this gradient? Because they actively transport H+ from low concentration in the companion cell into the cell wall. So this is known as step 1. It's active transport from low concentration to high concentration of H+. And this is active transport because the purpose is to constantly maintain a gradient between the companion cells and the cell wall. So this is where the ATP that is produced from companion cell is being used to actively transport the H+. So this will be against the concentration gradient. Then step 2. Once the H+ is accumulated in the cell wall, how does the H+ move down its gradient? You should be able to identify now that on the membrane here, you have a carrier protein. On the other side, you have another carrier protein. But this carrier protein, it's not an active transport. It's actually a facilitated diffusion because your H+ is going to diffuse down the concentration gradient back into the companion cell. So this is actually step 2, where it's actually a facilitated diffusion. And this is down the concentration gradient. Then as the H+ diffuse down the concentration gradient, that's where sucrose is being co-transported. But as sucrose is being co-transported, this is against the concentration gradient. So since it's against the concentration gradient, is it known as an active transport now? No, they are using the same carrier protein. So this is actually still considered as the facilitated diffusion, but we emphasize on the co-transporter. So when we said H plus diffuse down the concentration gradient, we will say that sucrose is being co-transported. And this co-transported, it's against concentration gradient. So why is it not an active transport? Because it does not involve ATP, and the energy that they use to move against the concentration gradient actually comes from the H plus. That means as a H plus is moving down the gradient, so it's like when the H plus is sliding down the gradient, they will actually drag the sucrose along using its energy. So sucrose is actually being dragged along against its concentration gradient. So when the sucrose is already in the companion cell, how does the sucrose enter the sieve tube then? So it will be easier now because there will be some pores on the cell wall, and the pores here are known as plasmodesmata. So the sucrose will just diffuse into the sieve tube through the plasmodesmata. So step 4 is just a diffusion. So you will see that in a simple loading of sucrose, it has involved 3 different types of transport. First one is active transport against the gradient. Number 2 and 3 is actually a type of facilitated diffusion, but one is down the gradient and the other is against the concentration gradient. And the last step will be diffusion through the plasmodesmata. That's why the whole process of active loading of sucrose is known as against concentration gradient, despite only the one step involves ATP. Because without the first step, which is active transport, step 2, 3, and 4 will not happen. So this is the whole idea of active loading of sucrose against concentration gradient. So now, after understanding this, see if you can apply to the question. So we go back to this. I'll refer to this question. Now, how sucrose is loaded into a sieve tube element? So it's like we know that there are like 4 main steps here. So always first thing, identify your companion cell and your sieve tube and your source. So here, this question, they only show the companion cell and the sieve tube element. So we know that sucrose is supposed to be coming from this direction to here. So now that means H plus need to be, the gradient need to be created at the source side. That means they need to pump the H plus from the companion cell out of the companion cell into the source cells. So since sucrose is over here, means my source must be over here. So number 1, you will start with H plus gradient created first. So this is number 1. Move out of ion into the companion cell wall. Actually, out of the companion cell, this is supposed to be into the cell wall. And this is actually against the concentration gradient. So this is definitely the active transport. So step 1, we will choose this. Then step 2, we know that once the gradient is created, H plus will need to diffuse down the concentration gradient where sucrose is co-transported. And this method is known as a facilitated diffusion. So don't choose active transport because it does not use ATP. So once the sucrose is already transported into the companion cell, step 3, how do they enter the sieve tube element? So they enter the sieve tube element is via the plasmodesmata. So it's a diffusion. So the answer is B. And as for the structure question, okay, so first thing, it's a simple chapter 4 question. It's just asking why transport protein are required to move sugar molecules such as glucose and fructose. So it's most likely related to the size of the molecule and the polarity of the molecule. So since they're water soluble and large, so they need a carrier protein to move them across the hydrophobic layer of the phospholipid. So the keywords will be, we'll talk about the features of glucose and sucrose. Large, water soluble, so they cannot pass through the hydrophobic core. Make sure you know this is the hydrocarbon chain of the phospholipid. Because this hydrophobic core is the one that is impermeable or barrier to water-soluble molecules. Then looking at this diagram, okay, so sorry, it looks complicated, but carefully identify where is the sieve tube, where is the companion cell, and where is the source. So what cells are they showing? So they say that sucrose is moved into the phloem sieve tube as shown. So where is the sucrose? So this is a companion cell. So they are focusing on the cell wall where you see there's lots of H plus here. So high concentration of H plus here. This must be the cell wall. So H plus is accumulated in the cell wall now. So obviously this side would be the companion cell cytoplasm, which has a lower concentration of H plus. So step 1, H plus is actively transported against the concentration gradient into the cell wall. So that's why another clue is, you see there's ATP used here, so to pump the H plus against the gradient. Then number 2, the H plus will move down the concentration gradient into the cytoplasm of the companion cell. And who gets transported? So that's where we will see that this is a clue that it's a sucrose. How can I tell? Because it shows there's 2 sugar units joined together. So it's a disaccharide. So this sucrose will be co-transported against the concentration gradient. How do I know it's against the gradient? Do you see here? I have high concentration of sucrose here. Here you have low concentration. So that's why this one is against the concentration gradient. That is step 3. So based on this diagram, they just want how the sucrose is moved. Okay, let's see which part the question wants. How they move into the companion cell, then how they actually move from the companion cell into the sieve tube element. So based on my diagram just now, you will be talking about step 1, 2, and 3 over here. And from the companion cell in the sieve tube element will be step 4. Yeah, it's 5 marks. So basically, you name the correct transport, you name the correct direction, and the gradient itself. I mean, whether it's against the gradient or down the gradient, you most likely get your marks. And then 2 examples of sink here will be, sink will be defined as cells that actually store glucose, store the sucrose, or use up the sucrose. So example will be like storage organs like root, it can be growing buds. So if your buds are growing, they need energy, or for your fruits, or for your flower that needs sugar for nectar, sugar in the fruit, so they can be a sink as well. Okay, so hope this explanation clears your doubt about how sucrose is actively loaded from the source into the sieve tube element. Thank you.
That's all for today's video. If you're interested in more genuine sharing by other geniuses, please subscribe to our channel and don't forget to turn on the notification bell, ding dong. Also, if you're struggling with 1 or 2 past year questions and the mark scheme just doesn't seem to help, Genie has got you covered. Feel free to let us know what question it is by filling in the Google form linked in the description below. Genius Hub will get genius teachers to fulfill your request for the solution. Genie, we'll see you next time. Bye-bye.